today we are going to begin with a eukaryotic cell. In our previous session, we have looked into all the fundamental insights of the chapter Cell, the Unit of Life, and today we are going to start with eukaryotic cells. We have also encountered the differences between a prokaryotic cell and a eukaryotic cell. Now, in a eukaryotic cell, we are going to learn about the organelles and the cytoplasmic constitution of both plant cell and animal cell. We will also look into the key differences between plant cell and animal cell, and then we will learn in detail about cell wall and cell membrane fluid mosaic model. So, those are the concepts that we are going to deal today, which are very important for the NEET and MCET as well, and knowing about the plant cell and animal cell in detail will help your understanding in ease of your understanding when you are dealing with single membrane bound organelles and double membrane bound organelles. Okay. So, first if we have to start with a plant cell, okay, a plant cell. We have discussed the limiting boundary of a plant cell is cell wall. Okay. Cell wall is freely permeable, it is not impermeable. Cell wall is permeable, and cell wall, the cells of the cell wall are connected to each other via plasmodesmatal connections. Okay. They are connected via plasmodesmatal connections. This plasmodesmatal connections will have the protein desmotubule, tubulin protein. Okay. Plasmodesmata are the connections in between the cell wall, which consists of the protein desmotubulin. This is about the outermost layer. We will learn what are the constituents of cell wall, what are the different layers of plant cell wall, we will look in detail. But as of now, remember, the limiting boundary is cell wall, which is freely permeable, and it is connected via plasmodesmatal connections. Next to the cell wall, we will have what? Cell membrane. Next to the cell wall towards the inner side, we have cell membrane or plasma membrane. About cell membrane and plasma membrane, we will learn in detail from fluid mosaic model. Fluid mosaic model. Got it? And then, now first we will look into the non-membrane bound organelles that are present in the plant cell. Now, only a membranous organelle, that is ribosomes. Okay, ribosomes are the only non-membrane bound organelle in a plant cell. So, if I have to take a plant cell, we have non-membrane bound organelles, the only non-membrane bound organelle is ribosomes. Where do you see these ribosomes in a plant cell? Ribosomes are present both in the cytoplasm of a plant cell, in cytoplasm of a plant cell, and inside the organelles like chloroplast and mitochondria. Okay. In cytoplasm, what type of ribosomes do you see in cytoplasm? In cytoplasm, you observe 80S ribosomes, wherein larger subunit is 60S, smaller subunit is 40S. Got it, ma? These are two different subunits of the ribosomes, that is 80S ribosomes, which are present in plant cells cytoplasm. Now, you see in chloroplast and mitochondria, you find 70S ribosomes in the chloroplast stroma and in mitochondrial matrix. You find 70S ribosomes where larger subunit is 50S and smaller subunit is 30S. Got it? Larger subunit is 50s and smaller subunit is 30s. This is about non-membrane bound organelles that you find in a plant cell. Clear, right? Only non-membrane bound organelle, those are ribosomes. You will see 80s ribosomes in the cytoplasm, wherein larger subunit is 60s, smaller subunit is 40s. Inside chloroplast and mitochondria, which are double membrane bound organelles, inside that only ribosomes are present in chloroplast, stroma and mitochondrial matrix. You have 70s ribosomes, 50s and 30s. Clear, Nama, till here? So, non-membrane bound organelle, that is about ribosomes, is clear. And at one more place you will find about find these ribosomes, that is over the surface of rough endoplasmic reticulum. Over the surface of rough endoplasmic reticulum. The ribosomes which are present over the rough endoplasmic reticulum are 80s only. So, where do you see these ribosomes? Inside the cytoplasm, which are freely present over the surface of rough endoplasmic reticulum, inside chloroplast, stroma and in mitochondrial matrix. What is the major function of these ribosomes? The major function of the ribosomes is protein synthesis. Synthesis of proteins. Protein synthesis is the major function of the ribosomes. Second is single membrane bound organelles. Single membrane bound organelles. Single membrane bound organelles. So, when you are dealing with single membrane bound organelles, you have to learn on the endomembrane system firstly. The extensions of nuclear envelope will form a reticulum of tubular structures. This complete network of reticulum structures are endoplasmic reticulum. Over those endoplasmic reticulum, where do you find ribosomes, are known as rough endoplasmic reticulum, and the ribosomes which are freely present without the endoplasmic reticulum, which is freely present without any ribosomes, are known as smooth endoplasmic reticulum. Remember ma, rough endoplasmic reticulum, as ribosomes are present, it is involved in protein synthesis. Smooth endoplasmic reticulum is involved in lipid synthesis. This is first single membrane bound organelle. And after this, the lipids and proteins which are synthesized by endoplasmic reticulum are modified and packed inside Golgi bodies or Golgi apparatus. Here you can see, right, this is one more single membrane bound organelle after ER, that is Golgi apparatus, where you can see complete packaging of the lipids and proteins. After Golgi apparatus, it is lysosomes. These are rich in hydrolytic enzymes, okay, which are known as suicidal bags of the cell. Okay. Next single membrane bound organelle is lysosomes. After that, it is vacuole. Vacuole is covered by a single membrane, and this membrane is addressed as tonoplast. Vacuole occupies over 90% of plant cells volume. Okay. So, these are the 4 single membrane bound organelles that you have to remember. Single membrane bound organelles, first is endoplasmic reticulum. We have 2 types of endoplasmic reticulum, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is involved in the synthesis of proteins. Smooth endoplasmic reticulum is involved in the synthesis of lipids. The lipids and proteins which are synthesized here are transferred to Golgi apparatus. Golgi apparatus. And here in Golgi apparatus, the lipids and proteins are completely packed. Packaging of lipids and proteins. After this, the packed lipids and proteins are released in the form of vesicles and these vesicles will be rich in the hydrolytic enzymes. These vesicles will be rich in the hydrolytic enzymes. Those are lysosomes. Lysosomes, which are rich in hydrolytic enzymes like carbohydrases, proteases, lipases and all. Next is vacuole, which occupies 90% of the plant cell's volume, and this vacuole is covered by the membrane which is known as tonoplast. Vacuole is surrounded by the membrane which is tonoplast. Got it right? Yeah. So, we have looked into all the single membrane bound organelles, which are rough endoplasmic reticulum, smooth endoplasmic reticulum and vacuole Golgi apparatus. Followed by this, we have cytoskeleton. Cytoskeleton includes microtubules, microfilaments and intermediary filaments. Followed by microbodies. Microbodies include peroxisomes, glyoxysomes and spirosomes. These also are single membrane bound organelles only. Now, looking into double membrane bound organelles, we have a nucleus, mitochondria and chloroplast. We have nucleus, mitochondria and chloroplast. Nucleus has 4 components inside it: nuclear envelope, nuclear pores, nucleoplasm and nucleolus. These are the 4 components of nucleus. Clear, right? And when we are discussing about double membrane bound organelles, we will discuss in detail about the components that are present in each double membrane bound organelle. Okay, ma? So, now if we have to look into the other single membrane bound organelles, other single membrane bound organelles include microbodies, and cytoskeleton structures. Then double membrane bound organelles. Includes, first is nucleus, includes nucleus. It has 4 components: nuclear envelope, nucleoplasm, nuclear pores and nucleolus. Right? And then we have second is mitochondria. Mitochondria also has 4 components: outer mitochondrial membrane, intermembrane space, inner mitochondrial membrane and mitochondrial matrix. These are the 4 components of mitochondria. Then when we have to look into the chloroplast, we have already learnt it in plant physiology, but for once again for our reference we will just recall it. Chloroplast is again a double membrane bound organelle, 2 membranes inside which, in between those 2 membranes we have periplastidial space, and inside the chloroplast we have thylakoids, which are membranous sac like structures. Stalk of that sac like structure is grana. One grana is connected to other grana via stroma lamellae. Okay? So, these are the constituents of chloroplast. I am not rewriting it because we have already discussed this in detail in plant physiology. So, here what are the organelles that are present in a plant cell? Non-membrane bound organelles, ribosomes which are present in cytoplasm and over RER, which are of ATS type of ribosomes, Chloroplast and mitochondria, inside them we have 70S type of ribosomes. These are non-membrane bound. Single membrane bound endoplasmic reticulum, Golgi, lysosomes, vacuoles, microbodies and cytoskeleton. Double membrane bound organelles, nucleus, mitochondria and chloroplast. This is the complete picture of a plant cell, okay, from which what are we going to learn in detail today is cell wall. About plant cell wall we are going to learn completely. Before moving into a plant cell wall, let us also discuss about and the labelings of this diagram are very important, each and every label, remember that carefully, okay. First let us start from the center and move to the complete organelles. At here where you can see the nuclear envelope with pores and genetic material at the nucleolus here inside, this is nucleus, and the network of the reticular structures which are above the nuclear envelope are what? Endoplasmic reticulum. If you can find small ribosomes over the ER, just go for the marking of rough endoplasmic reticulum. Next to it, if the sausage shaped structures are there with the cristae, inner membrane foldings, then you mark it as mitochondria. Then these green color plastids are marked as what? Chloroplast, small microbodies, and this cisternae like structures which are present, disc shaped cisternae structures which are present are what? Golgi bodies. 90% of the space is occupied by this large organelle, that is vacuole. Got it? These are the major labelings that you have to remember with respect to the diagram, because that is a standard NCERT diagram and it is very important. We may expect in diagram based questions. Next, about an animal cell. Just a very few minute changes, rest part will be same as that of plant cell, okay. So here if you recall carefully, the outermost membrane is what? Plasma membrane only, here cell wall is absent. Plasma membrane is selectively permeable membrane, okay. It is selectively permeable membrane. It is selectively permeable because it is selective in the choosing whether to allow few solute particles or not, okay. So it is selective in permeability. And then next to the plasma membrane, if you see the cytoplasm constituents, first non-membrane bound organelles include two here, one is ribosomes and second is centriole. Centrioles are non-membrane bound organelles. These are two cylindrical structures which are present perpendicular to each other in a centrosome, okay. First non-membrane bound organelles in an animal cell. First non-membrane bound in an animal cell. First includes ribosomes, second centrioles. Fine, right. Ribosomes also we can see of two types, one is 70S and the other is 80S. 70S ribosomes are present inside the mitochondria. 80S type of ribosomes are present over endoplasmic reticulum. They are present over endoplasmic reticulum and they are present inside the cytoplasm freely also. Got it? These are the locations of different types of ribosomes. Centrioles are two cylindrical structures which are arranged perpendicular to each other, whose major role is in formation of spindle apparatus. About spindle apparatus you will get a clear idea when we are discussing in cell cycle and cell division, okay ma. So centrioles are two cylindrical structures which are perpendicular to each other. They are present inside a centrosome. A single centrosome will have two centrioles. Got it? Their major role is what? Their major role is spindle apparatus formation. Spindle apparatus formation. Got it? These are non-membrane bound organelles. Next, coming to the single membrane bound organelles, it is same as that of a plant cell, but there is one difference. In plant cell you can see vacuole which is occupying 90% of plant cell's volume, but here in the animal cell vacuoles are very limited or absent. Okay, vacuole number is very limited or they are absent in the cells, because animals are locomotory, plant cells are non-locomotory. Here if you can observe, rough endoplasmic reticulum are the reticular structures above which you can find the ribosomes, RER, and then SCR in close proximity to this, the same tubular structures arranged in reticulum in close proximity to RER, followed by peroxisomes, Golgi apparatus, you have lysosomes. All these are single membrane bound organelles only. Coming to double membrane bound organelles, you can see here nucleus. Inside nucleus, nucleolus is present and it is covered by a double membrane bound nuclear envelope. Same organelle, single membrane and double membrane organelles are one and the same. The only difference is encountered in vacuole presence. Vacuole is occupied 90% in a plant cell and it is present in the minute vesicles or limited in its number in an animal cell, because they are locomotory. Got it? So, this is all about an animal cell. Major point you have to remember, that is about centrioles. Centrioles are the non-membrane bound organelles which you can observe only in the animal cell but not in a plant cell. Got it, ma? In plant cell the only non-membrane bound organelles are ribosomes. Quite a times you got question on the centrioles itself, very important to remember. Okay. And then these are the major differences in between a plant cell and animal cell. Then we can move into the clear analysis of the cell wall. First, cell wall is present in a plant cell. We will look into the components, but the major composition is of cellulose. Cell wall is absent in animal cell. The shape of a plant cell can be regular in shape or rectangular. This is irregular or round in shape. In an animal cell, the cells can be irregular or round. Plastids are present in a plant cell. Plastids not just include chloroplast, it also includes leucoplast and chromoplast. Leucoplasts will act as storage bodies. Chromoplasts serve as, they will give the color to the pigments. These are present in all the plant bodies and plastids are absent in an animal cell. And as I told you, the major difference comes with a single membrane bound organelle, one organelle, that is vacuole. Vacuole, you can see large central vacuole with the membrane tonoplast which occupies 90% of the plant cell volume in a plant cell, and absent or small or temporary, limited in its number in an animal cell. Centrioles are absent in a plant cell, centrioles are present only in an animal cell. These are non-membrane bound again. What is the major function of centrioles? The major function is formation of spindle apparatus. Next is lysosomes, which are again single membrane bound organelle. Lysosomes are rare in a plant cell, they are present in abundance in an animal cell. You can come across different types of lysosomes like primary lysosomes, secondary lysosomes or heterophagosomes, tertiary lysosomes and residual bodies. These are different types of lysosomes. We will learn in detail in our academic session. Next, mode of nutrition is purely autotrophic in plants and it is purely heterotrophic in animals. Cilia and flagella is rare in plants. Cilia and flagella are often present in animal cells. Cytokinesis is by cell plate formation. Okay. How cytokinesis is happening in a plant cell, we will also see that, and this forms via cleavage furrow, cell division. Cytokinesis is via cell plate formation in a plant cell and it is via cleavage furrow in an animal cell. Okay. And then storage food is starch in a plant cell, it is glycogen in animal cell. Okay. Glycogen in animal cell and starch in a plant cell. Clear, right? Now we will move on to a cell wall. First, Before looking into the cell wall, I have to introduce two kingdom system of classification, because only in two kingdom system of classification there is a chapter that is biological classification you will learn, wherein the first phase of the chapter is about kingdom systems of classification. In kingdom systems of classification you will come across 2 kingdom, 3 kingdom, 4, 5 and 6 kingdom systems of classification. So, the initial 2 kingdom system of classification is done on the major criteria, that is cell wall. Okay. 2 kingdom system of classification whose major criteria is cell wall, wherein 2 kingdoms that are divided are Plantae and Animalia. All the organisms, be it prokaryotic, eukaryotic, autotrophic or heterotrophic, unicellular or multicellular, all sort of organisms, if they are having cell wall, they are placed under Plantae. If they are not having cell wall, they are placed under Animalia. So, those were the major drawbacks of 2 kingdom, but when it comes to the kingdom Plantae in 2 kingdom system of classification, fungi is included in Plantae, algae is included in Plantae, plants of course they are included under Plantae, even bacteria is included under Plantae because it has cell wall. Okay. So, learning the compositions of all the cell wall is very important. Fungal cell wall is made up of chitin. Fungal cell wall is made up of chitin. Chitin is known as fungal cellulose. Chitin is known as fungal cellulose. This is a polymer of NAG units, which is N-acetyl glucosamine units. NAG units are N-acetyl glucosamine units. Algal cell wall, it can be made up of galactans, mannans, minerals like calcium carbonate. Okay, it can be galactans, mannans, minerals like calcium carbonate. Plant cell wall is made up of cellulose, which is insoluble polysaccharide and which is a homopolymer only, homopolymer of glucose. Cellulose is a homopolymer of glucose. Cellulose is a homopolymer of glucose, and along with cellulose, hemicellulose, pectins, proteins are present. These are the compositions of plant cell wall. And bacterial cell wall is made up of peptidoglycan. Bacterial cell wall is made up of peptidoglycan. All the compositions are very important. Bacterial cell wall composition, peptidoglycan. Fungal cell wall is chitin, which is of NAG units, that is N-acetyl glucosamine. Algal cell wall, galactans, mannans, minerals like calcium carbonate. Plant cell wall are insoluble polysaccharides like cellulose, hemicellulose, pectins and proteins. Cellulose is a homopolymer of glucose. Clear, right? These are the compositions of different cell walls. Now, when it comes to plant cell wall, how at the beginning the cell wall is formed. Just now we have seen about cytokinesis of plant cell, cytokinesis via cell plate formation. Cytokinesis is via cell plate formation. Yeah, good afternoon. Please continue with the session, ma. I have seen your message, but there is no doubt in that. First, we have seen the composition of a plant cell wall. Here, yeah, plant cell wall at the beginning, if this is the cytokinesis that is happening, at the center first there is a small layer which is developed, which is rich in, this is rich in calcium pectate and magnesium pectate. Calcium pectate and magnesium pectate. This layer is known as middle lamella. This layer is addressed as middle lamella. And next, when this cell plate, when this middle lamella is extending towards the periphery from center, okay, center a small layer is formed that is known as middle lamella. This is rich in calcium pectate and magnesium pectate, and which organelle is synthesizing this middle lamella? This is from Golgi bodies or Golgi apparatus. It is synthesized from Golgi bodies or Golgi apparatus. From the center, this is moving towards the either of the peripheral ends. This is moving towards the peripheral ends, and when it is moving to the peripheral ends, at the end it forms a cell plate. Cell plate. Fine, right? Cell plate is formed. Once it is moving to the peripheral end, cell plate is formed. Clear, right? So, this from center it is moving to the periphery. So, the formation of cell plate is centrifugal. Centrifugal formation of cell plate takes place in a plant cell wall. Clear, right, ma? Once again I will repeat, at the beginning, at the center, okay, during the cytokinesis after telophase, what happens? At the center, middle lamella is formed. This middle lamella is rich in magnesium pectate and calcium pectate. These are synthesized from Golgi bodies. Once this is formed at the center, this moves towards the periphery, forming out what? Cell plate, and this mode of formation of cell plate is known as centrifugal method of formation of cell plate. Once the cell plate is formed, middle lamella, right? So, the first initial formation of the cell plate is what? Middle lamella. Middle lamella is the first formed layer in between two plant cells. In between two plant cells, middle lamella is the first formed cell layer. After middle lamella, next to the middle lamella, what is formed? Yeah. Here we go. We have to suppose these two are plant cells. Plant cell walls. At the center, this is the plasma membrane, okay, and these are the organelles which are present inside. In between two cell walls, the first layer which is formed is middle lamella. This is the common wall between two plant cells. The common wall between two plant cells is middle lamella. Next to the middle lamella, towards the cell membrane, next to the middle lamella, towards the inner side, the first layer which is formed is known as primary cell wall. Okay. Next to the middle lamella, the first cell wall layer which is formed is primary cell wall. Primary cell wall is thin, it is extensible, it is made up of cellulose. Okay. Primary cell wall is thin, extensible, and it is made up of cell wall. Next to the primary cell wall, towards the inner side, secondary cell wall is formed. Next to the primary cell wall, towards inner side, secondary cell wall is formed, and the secondary cell wall can be of 2 to 3 layered. Secondary cell wall can be of 2 to 3 layered. Secondary cell wall is 2 to 3 layered. Got it, ma'am? So, now after the formation of secondary cell wall, if you observe clearly, close to the cell membrane, what cell wall layer is there? Secondary cell wall. Next to the secondary cell wall, you have primary cell wall. Next to the primary cell wall, middle lamella is there. So, the hierarchy from peripheral end to the center, if you see, the hierarchy from peripheral center to the periphery to the center, first is middle lamella, next to the middle lamella it is primary cell wall, next to the primary cell wall you have secondary cell wall, and then plasma membrane. This hierarchy is very, very important, okay. And With the increase in secondary cell wall layers, at the center, the wall layer which is next to the secondary cell wall, that is primary cell wall, can get disintegrated. It can lose its structural stability with the increase in the secondary cell wall layers, because it is thin and extensible. This is all about cell wall of a plant cell. I hope you are clear with it. Fine, right. So, with this we are done with the cell wall of a plant cell. In our next class, we will look into the cell membrane in detail, and then we will also look into cell cycle and cell division. Thank you.